Good evening, aloha. Let me first thank to Hawaii Sea Grant and Hanauma Bay for giving us this opportunity to be here tonight with you and share our ideas and share our knowledge and experience in the field of coral conservation. Sorry. So to briefly present us, we are on the other part of the island. We work and we have our lab in the windward part of Oahu in Kaneohe Bay. So here in these pictures, you can see overview of Kaneohe Bay with a zoom in on Coconut Island. So this is where Hawaiian Institute of Marine Biology is located. And you can see that it's literally encircled by coral reefs. It's a great place to study corals. Kaneohe Bay is a perfect place for these patch reefs to be formed. They are overgrown by corals and they are doing quite nice. So it gives us the opportunity to study them in the bay, in situ, to be very close to corals, and we have this close. We can access the living library of corals any time of the day, any time of the year, and that's perfect. So if you take your mask and you go snorkeling, you dive into waters of Kaneohe Bay, this is basically what you are going to see. It's pretty much everywhere. If you are lucky, you will encounter some turtles, and from time to time you can encounter a group of scientists. There are some reefs where we have our underwater benches, underwater tables, where we outplant some corals, and we can study them. We have tags on them. We know exactly their history. We know how they behave. We know everything about them, and we can study them directly underwater. If you go closer to coral reef, what are you going to see? So people in the past, they believed that coral reefs are made of rocks. Then they thought, when they saw that it's a living thing, so they thought it's plants. But if you look really closely, you will see that every coral is made of these little tiny polyps, and it's hundreds, it's thousands of them. It's actually clones. One is exactly the same as the other one, and they form one coral. If you look very, very close on this polyp, you'll see that it's actually an animal. It has a mouth. Here it has tentacles with stinging cells, and it can hunt. It has these stinging cells that it can kill little tiny prey, and it can put it in its mouth. It's definitely an animal, but beside being animal, it has these very little and very tiny spheric organisms living inside of its tissue, living inside of its cells, and it's symbiotic plants, it's symbiotic algae. And this symbiosis between animal and plant is crucial for the survival of coral. Why? Because coral provides shelter and provides CO2 to algae. And since algae is a plant, it has the gift of photosynthesis. So if a plant is given CO2 and energy from sun, it can create sugars, which is energetic source, and it creates oxygen as a byproduct, a very important byproduct in our world. So coral can get its nutrients and its energy from algae that live within its tissues, and algae gets shelter, so it's safe from any predators and from any herbivorous organisms around. But unfortunately, this symbiosis can be disrupted. So what happens? You might have heard about coral bleaching. So this is what happens when the symbiosis between coral and algae is disrupted. Why do corals bleach? Because these plants living in the tissue, they have chlorophyll, they have many pigments, they have many substances that are colorful, and they give the coral its color. So when these organisms decide to leave coral, or when they are expelled, or whatever happens with them during stress, corals bleach. And while they are bleached, it means that they don't have their energy, their power plants, they are missing. So they can't really get nutrients for them. They start starving. And if this moment takes much longer, then the coral can survive with its inside energy storage, it will die. So that's why coral bleaching is very bad event that can happen mostly when it's massive coral bleaching, when there is whole coral reef that is bleaching. It can lead to very rapid death of all the reef. And why I'm talking about this is because we are actually seeing this. you may ask, like, why is coral bleaching happening? What makes these little symbionts leave? So there are multiple reasons, but the main threat and the main reason why corals bleach is because of temperature, high temperature stress. So this has nothing to do with, or it's not that the temperature of sea is like very gradually increasing. This is not what makes corals bleach. What makes them bleach is the sudden temperature stress, high temperature stress. So you have a tropical storm that comes, you have hurricane that comes, you have some extreme weather coming, the winds turn, suddenly there are no currents, and there is accumulation of hot and warm water around coral reefs. And this makes them live in very stressful conditions. It makes their symbionts leave the coral, and corals die. So here in this picture, you can see main global bleaching events that happened. First one in 1998, and then two global bleaching events in 2015 and 2016. When I say global bleaching events, I really mean global. So it happened all over the world. Almost every coral reef in the world had some portion of corals that bleached during these, at least part of these 2 years. So it was massive event where we lost between some 25 to 70% of corals, depending on particular reefs. So this was very sad 2 years, 2 consecutive years. What about here in Hawaii? So you can see a graph of temperatures in Kaneohe Bay, and you can see that there is a threshold of 30 degrees Celsius. And there were 3 events, 3 years when the threshold was broken, and it was in 1996, 2014, and 2015. So during these 3 years, we could see bleaching events, massive bleaching in Kaneohe Bay. Luckily, this event did not last long enough to kill all these corals, so the mortality was around 5%. But it leaves us with the big question. So when is the next bleaching event? When is it going to hit next? So right now, in this moment in the world, there are 3 places where massive coral bleaching is occurring. Here you can see one in Thailand, in Patani. The second one that has been occurring for maybe 2 months already now is in Moorea in French Polynesia, where they see a massive coral bleaching. These are real pictures taken underwater right now. The third one is in Sri Lanka, where apparently over 90% of corals in the eastern part of Sri Lanka are bleached. Unfortunately, I couldn't find any pictures of these. We have only reports. And you can see that NOAA has already issued a coral reef watch alert, let's say, for approaching summer season. And we are seeing in Kaneohe Bay, we are seeing some very elevated temperatures right now in day peaks. So we are worried that next bleaching event could strike now. So as you can see, this is not a problem that might happen in 20, 30, 50 years if we are not doing anything. You know, it's usually people, when they talk about climate change, they are like, if we don't do anything now, in 20, in 30 years, there might be a problem. This is something that is happening right now. So we have corals in the world that are dying right now, and we need to do something to save them. Climate change-driven warming events are the main threat to coral reefs worldwide right now. So in the Gates Lab, we investigate solutions such as making corals more resilient to sudden thermal stress. And our aim is to buy us some time, you know, to know that now we can do something to help corals survive 10, 15, 20 years, to buy the time before like real measures are taken. Kira is going to talk about this in more details in the second part of the presentation. So when I joined the Gates Lab a year and a half ago, just before I was working in cancer field, I was trying to understand why cancer cells become immortal and how it happened. Then I decided to change to coral reef science, but I'm still sort of asking myself the same questions. Why do they die? What makes them die on cellular level and molecular level? And also, I was just wondering and asking myself the question, can corals acclimatize to withstand thermal stress from warm water events? And why did I think so? Well, we have examples of acclimatization everywhere around us. Where I came to Hawaii, first day, my skin got sunburned. Of course, I was not used to it. I come from Europe. The sun exposure is totally different. But when I just stayed on the sun for a very short period of time daily, after a week, after 2 weeks, I stopped being sunburned. I know this is temporary. If I leave to Scandinavian countries now and I stay there for a month, I come back and I will get the sunburn again, of course. But my skin was able to acclimatize. The same thing would happen to me if I started climbing up. You know, if you go to Mauna Kea or Mauna Loa, they will tell you to wait in the like basic camp, I like to call it, for 20, 25 minutes to a little bit acclimatize. If you decide to go to Mount Everest right now, you'll probably die. But if you stay 2-3 weeks somewhere in lower altitudes, you can survive this. So, and there are many more examples. So I asked myself the question, can corals have the same mechanisms? Can they acclimatize to heat stress? So my preconditioning experiment was, okay, if I take a coral and put it to sublethal temperatures, I will let it time to learn how to cope with high temperature stress, but it won't kill it. So it will be a temperature that will stress it, not kill it, and it will have time to learn, to understand how to behave in these conditions. So for this, I took 8 colonies of Pocillopora acuta. It's this little cute coral. I divided every colony in 2 halves, and 1 half I put to 29 degrees Celsius for only 3 days, very short preconditioning. Then I returned it back to 26 degrees, which during winter is ambient temperature. The second half of the colony remained at 26 degrees. I waited 2 weeks, and then I pretended that there is like a storm or something coming, bringing very warm water of 32 degrees. And I wanted to see what happens in terms of bleaching. So you can see that for non-preconditioned corals, after 3 days, they bleached. But preconditioned corals, after 3 days, you could not really see the difference between preconditioned and control corals. Well, I'm a scientist, so I wanted to quantify it. I asked myself a question, how to quantify this bleaching. So we can quantify it using confocal fluorescence microscopy. I'm not going to go into details, just believe me that fluorescence microscopy is something that allows us to visualize different tissues and different cells in different colors. So what I could do was to visualize host cells, coral tissue, and coral cells in green. I hope you can see at least something. And there are the plant cells that have a lot of chlorophyll. So I could visualize chlorophyll under microscope as a red signal. So then by combining the picture, I could simply analyze the ratio of red to green. So when coral was bleaching, it was losing the algae. So the red signal was getting lower and lower. And by quantifying this, by analyzing this, I could quantify the bleaching rate. So here you can see an example of non-bleached coral in the left, with very strong red signal, and bleached coral with almost no red signal or very low red signal. So it worked, and when I put this in graphics, I could definitely see that the preconditioned corals depicted here in gray had much slower bleaching rate than non-preconditioned coral. They would eventually bleach as well, probably, I think maybe in many, many days, but it was slower. So that was a success. And since I'm molecular biologist, I was curious to know what is the mechanism, what's happening in the cell or on molecular level. So we know some coral bleaching mechanisms. We don't know much about it. We just know that they exist. Somebody has seen them. Somebody has written a paper about them and set like some basic characterizations. But we have not many knowledge in this field so far. There are multiple ways how corals can bleach, but I was mostly interested in these two. It's symbiophagy. It's a moment when a host cell, so coral cell, will lyse the symbiont within its cell and will probably reuse the coral, the algal cell for its own purposes. And then host cell apoptosis. Host cell apoptosis is also called cellular suicide. It's a moment in your body when, or in like bodies of all of us, when a cell gets a signal. It can be like, okay, something is going wrong. I should not live here. I should not be here anymore because I'm dangerous for my environment. I will kill myself. And then in a very neat, programmed way, the cell will kill itself. So we know these pathways. pretty well from humans, from mice, from worms. They are very well studied. And fortunately for us, they are very conserved across the species. So I could use this knowledge in studying corals. So here, just for you to see, they are very, very complicated. These pathways, they interact together. They use the same genes at some parts. Like, I'm not going to distinguish between these two pathways. I'm just going to refer to them as cellular death pathway. The important thing is for you to imagine that it's a complex interplay of pro-life and pro-death proteins. So there is a signal coming from the outside of the cell, and then the cell responds. And imagine it as a fight between pro-life and pro-death proteins, and the side that wins will determine the fate of the cell. So it can decide between death and life, just based on this pathway. So I know I picked few genes, most importantly BCL2, which is a very well-known pro-life and very strong pro-life gene, and bugs that are very strong and known pro-death genes. And I was analyzing what happens with the ratio of these pro-life and pro-death within the first 24 hours of heat stress in preconditioned and non-preconditioned corals. So here you can see that between 3 and 6 hours, in gray, it's preconditioned corals. There is an increased ratio, there's a shift in the ratio of the pro-life versus pro-death genes, but in favor of pro-life genes. So it seems that in preconditioned corals, the pro-life side is winning. I'm saying it very simplistically, but I hope that it's clear for you. So this was the observation, but then we need to know whether this shift is what causes corals not to bleach. So we know that it is happening, but we don't know if that's the thing that makes them more resilient, more tolerant to stress. So you know that there is like an increase ratio that maybe slows down the bleaching rate. So how to analyze, how to study if that is the causing thing? Well, if you decrease the ratio back to where it was at the beginning and you lose the effect, you lose the phenotype, then you can be pretty sure that yes, that was the causing thing. So I used inhibitor of this BCL2, so I know that in preconditioned corals there is higher ratio of pro-life gene, so I put it back. So I blocked that pro-life gene, and I was studying what happens. And in my theory, I thought if I block this gene in preconditioned corals, and if this gene was helping coral to be stronger, then by blocking it, the coral will no longer be stronger, and suddenly it will behave like if it was never preconditioned. And that's exactly what happened. So here you can see that the control, preconditioned and non-preconditioned corals, you already saw that. And here again, this is the preconditioned coral that was not bleaching after 3 days. But as soon as I added the inhibitor, it lost its strength. It became again like non-preconditioned. Here, you can even see it in graphs if you did not believe my picture. Even the graphs show that here, without any inhibition, there was a clear difference in bleaching rate. But as soon as I applied the inhibitor, the effect, the phenotype was lost. So now we can say, okay, so the BCL2 inhibition reversed the higher thermal resilience. So in summary, we can now say that rapid thermal preconditioning slows down the bleaching rate in Poecilopora acuta, and this happens through the programmed cellular death pathways. And more importantly, we know that the pro-life genes are very important in this process, and it's basically through increase of the pro-life genes that the corals can be more tolerant to heat stress. So of course, the next question that we are asking now is, can we mimic the preconditioning and make corals more thermally tolerant before a warming event hits the reef? So that's a big question. We are currently working on that. It's already good to know that we can modulate these pathways and we can make coral bleach when we want. So now the second point is, could we somehow help corals not to bleach if we know that there is a heat stress and heat like warm event that is coming? So Kira is now going to tell you the responses on these questions and how we try to manage this restoration process. Thank you.
Thank you, Eva. So it's pretty amazing that coral bleaching is such a recent phenomenon that we don't even fully understand the mechanism. So that's why we had to bring someone out of cancer research in order to really investigate what is actually happening when corals bleach. So what I'm going to do is zoom out a little bit and tell you more about what our lab is doing as a whole team, and also kind of where we're headed. So again, just to recap, I think we all understand the problem. As Eva said, it's not this gradual increase in temperature. We've shown that corals can naturally acclimatize, and we want them to be able to do that. It's these sudden events that all of a sudden come in and put them under this thermal stress and just pretty much cook them because they're benthic organisms and they can't move and they can't escape it. So what's really special about our lab, and we've been recognized since the beginning, where we were led by Ruth Gates, who passed away recently, but it's her amazing optimism. So yes, we see a problem. This change is occurring faster than corals can adapt, but you know what? We can do something about it. We can have hope and at least buy us some time until we can get a handle on greenhouse gas emissions, because that's the real big threat. Don't worry right now, don't worry about sunscreens, don't worry about all that kind of stuff. Our threat is greenhouse gas emissions. If we don't get a hold of that, nothing else is going to matter. So the solutions that Ruth and also Dr. Madeline Van Oppen from Australia Institute of Marine Biology came up with all the way back in 2013, before some of our major bleaching events here, was to build this biological toolkit that will allow us to do assisted evolution. So assisted evolution is a pretty common term now in the biological field. But at the time, it was really only used kind of in one study in the terrestrial system. So we brought that out of the terrestrial system and applied it to the marine system. And the idea is just to give a little bit of a push to accelerate those naturally occurring processes that the corals usually are going through to naturally acclimatize. So within this biological toolkit, it was really like this crazy idea of assisted evolution. So no one had ever tried something like this to actually intervene to help corals survive. And so it was really just a proof of concept study. So they came up with these 3 methods that are crazy, but they might work. Eva has already gone through inducing acclimatization. So that's like conditioning, like she said, you're kind of training them so that when they are actually exposed to the real threat, they can be ready for it. Selective breeding is another method, and also modifying symbioses. And you might have heard of this as the supercoral project. The uptight scientists only like to use assisted evolution, but supercorals are pretty fun too. So I'm not going to go too much into inducing acclimatization because that's what Eva just went through. But as everyone know the saying, what doesn't kill you?
Makes you stronger.
Yep, makes you stronger. So that's the main idea behind it. So that's her. I gotta use this. So this is Eva's example from her experiments, and then I just, she was using heat to precondition. I did want to point out a study from one of our PhD candidates, Ariana Huffmeyer, where she actually looked at using. I guess I could use. Using cool temperature. So what happens if you precondition them with cooler temperatures so that, then they can gain these nutrients and be kind of in a better state of mind right before they go through the stress. And she did see differences with that as well, and is still going to investigate it a little bit farther. And she also coupled that experiment with nutrition. So remember, the main reason when corals bleach that they might die is that they're losing their main food source, which is in the algae. They're producing those sugars through photosynthesis. But they can also, as Eva said, heterotrophically feed or feed with their nematocysts, their stinging cells, which is zooplankton that's in the natural water environment. So she coupled those. And as you can see, this is one that was fed and in a cool treatment. And this is one that was unfed and in a, I'm actually not sure which treatment this one was in, but as you can see, the colors are very different. When you do feed these corals, they're getting this nice deep pigmentation. But I think this one was just in ambient water, not in the cool treatment. So the second kind of proof of concept idea was selective breeding. So I'm sure most of you know because you come to these talks regularly, but corals can sexually reproduce. So they can have eggs and sperms depending on the species. They also asexually reproduce through brooding, or where they just make clones of themselves, or also fragmentation, where they just break off and they can start a new, the same colony, the same clone, but start in a new place. So selective breeding is looking at sexual reproduction, and it's intentionally breeding two parents to get a specific trait out of it to be passed on to the offspring. So in this case, what will we be looking for? Which trait? What's our biggest threat right now?
Bleaching.
Bleaching, right? From temperature stress. So what trait do we want our offspring to have? Yep, tolerance to heat. So we want our offspring, we want to be able to breed two parents that have traits that show that they are more thermally tolerant to start. So those are corals that did not bleach in these natural bleaching events, and see if our offspring can be even stronger. So we've looked at two species with this type of approach, and one is Montipora capitata. They're actually hermaphroditic, so they release these bundles that float to the surface that have eggs and sperm in them, and then they break up and they can recombine with eggs and sperm from other colonies in the water column, but they won't self to themselves. It's a very low chance. And then there's Porites compressa. So these are the two main reef builders in Kaneohe Bay. We have a lot more diversity here at Hanauma Bay. If you go in Kaneohe Bay, like, you mainly just see these two species. But you can have 100% cover in some areas, which is amazing. And they're typically a little bit healthier than over here for different reasons. But Porites compressa is gonochoristic. So they have male and female colonies. So one releases sperm and one releases eggs. So different methods, but again, we're trying to answer some of these questions about whether these offspring can be stronger and whether we can raise those and do it at a scale that's large enough to actually make an impact. So this is Beth Lenz's project, and can just quickly show you, we have to use an army of volunteers when we do this because it's a lot of work. But up here you see some of the egg bundles before they actually get fertilized, and this is after fertilization. You can also see this is what volunteers do for us. They collect these egg bundles, and actually we invite all volunteers from all over to come and help. So you're welcome to get in touch with us. But she is looking, one, at the impacts of bleaching history on offspring performance. So knowing the history of the parents, and how that translates to their offspring, and actually like how fecund those parents actually are. And also preconditioning these parents, just like how Eva did, putting them in this temperature stress, but for over a long period of time, about 3 months while they're going through gametogenesis and developing their eggs and sperm, and looking if they're preconditioned during gametogenesis, if that then benefits their offspring. So again, a big issue with this is scaling up. So we actually go out into the field. We've done this the last couple of years, and we net the colonies. We have these colonies tagged out in Kaneohe Bay. And because Montipora capitata said the egg and sperm bundles float, we can just let them go ahead and release, and then we catch them in this Tupperware bucket and then screw on the cap. We can bring them back to the boat, and then we know which colony they came from, and we can go ahead and mix them into buckets. So what we do is the ones we netted are the ones that did not bleach in 2015, that bleaching event, when we were ready for it and we had tags and we were able to tag them. And then we compare them to wild types. So that's just collecting random gametes from around Kaneohe Bay and see when these grow up, which ones are stronger. And then our Australian partners, I just wanted to mention, are doing something really cool that we don't have the ability to do here, because they can form hybrids with Acropora species. So that's the same genus, they have different species, and they can recombine those to form hybrids. So this right here is a purebred in temperature stress, and this is a hybrid. So you can see they perform much better than the purebreds. The reason we just don't have enough diversity here to be able to do that. So the idea with bringing Australia and Hawaii together in this project is to do some parallel experiments and see how does that differ with what Australia has, what species they have, and what we have here, and also access to the field and different benefits that we each have. So the third proof of concept that we were looking at is modifying symbioses. So coral is a really complex organism. This is what Eva was already talking about. This is a bleached coral, so that you see there's barely any green. And you see just a few, sorry, you should be able to see the green, but you see just a few of the red dots because it's a bleached coral. But with that, we also have bacteria, you have viruses, you have all sorts of things that are interacting with the coral. So we're looking at, one example is, I'll just talk about algae. So if the algae is more thermally tolerant, then it might not go into overdrive when the heat stress is there. So then the coral won't have to release it as quickly as it would for another type of algae. So if you can have the coral pick up that type of algae that's more tolerant to thermal stress, then maybe that coral won't have to bleach at all. So that's the idea, is kind of switching out the algae partners that it has with more thermally tolerant ones. Here in Hawaii, we have all vertical transmitters. So our coral is born with algae already in its cells. So you have to actually experimentally bleach that coral and keep it alive in order to inoculate it with new algae. In Australia, another benefit of partnering with them, this picture is actually taken there. You can actually, some of their species are born without algae. That's their horizontal transmitters, and they pick it up from the environment. So it's pretty easy to just go ahead and give them the algae that you want them to take up, and then stress test them and see if they'll actually be stronger. You can also modify the other symbionts that I was talking about, bacteria. You could do probiotics. There's all sorts of new ideas like that. So this is just an example. I'm not going to go too far into it, from a collaboration we did with Australia, where you could look at preference of uptake. So maybe these are all different types of algae. And as you can see, there was a lot of uptake of D1. It didn't necessarily mean anything yet. The main point of this story was there was one that it was, that one they only were allowed to take up those. And then this one, they were allowed to have a choice. So it's a choice experiment. And then now we're testing to see which ones those larvae actually took up, to see why that preference exists, and is it going to prefer one that has thermal tolerance? Would they know that? So our main goal with all of this assisted evolution research was to be able to implement this. I'm just going to check time really fast because I want you to be able to ask questions. Okay, we're doing good. So like I said, assisted evolution was this new crazy idea, and we just got funded for it 4 years ago. Someone, Paul Giallen Philanthropies, took a chance on us when no one else would, and just said, yes, this is crazy, but let's try it. And we've seen every single one of those methods. We've seen that we've been able to make a difference. However, we're not ready to go ahead and say, all right, let's put all these corals out in Hanauma Bay and just see what happens. Because we are altering things. We're aware of that. We're not doing GMOs. We're not genetically modifying them, but we are having some kind of intervention with them. And before we do that, we need to look at trade-offs. So like we talked about, traits of thermal tolerance. Maybe if you select for that, that's great for thermal tolerance, but what are you missing then? Are there trade-offs? Are there other things that those corals are lacking? They might still not be the best performers for other reasons that we don't know yet. We just haven't been looking at it long enough. Another trade-off, or another thing that we need to investigate before we put these corals into the field, these super corals out, is whether or not things are changed with multiple generations. So we haven't had enough time. Corals take quite a while to get reproductively active, to get old enough to be able to reproduce, the sexually reproducing ones at least. So we need a little bit more time to look at those next generations and see how those trade-offs are impacting them. So this is definitely not in Hawaii. This is the Biosphere 2. I don't know if you've ever seen the Biodome movie with Pauly Shore, but it exists. And let's see if I'm a cursor. So we can try all these crazy mad scientist methods at the Biosphere 2 because it's all the way in Arizona and not anywhere near an ocean outlet. So we're not concerning anyone by doing all sorts of things. So we've talked about bringing in quote unquote super corals from around the world, putting them in their big pool, which is over here, and seeing how they interact, seeing how this community system actually works. And so we just really need to make sure before we start implementing these ideas with supercorals, whether that there's any risk, if it's even cost effective to do these methods at a large scale, and then also whether it will actually improve reef prognosis. So now we are going to still move forward with implementation because there's something else that has been really important through this research the last 4 years. And that is that we are able to identify resilience. So I've talked a lot about this living library that we have down here. So during the bleaching event in 2014 and 2015, but more so in 2015 because we were prepared, we tagged these corals. So this is a bleached coral right next to one that didn't bleach. So our main question is, why is that happening? What is the benefit that this one has to not bleach? But now they look exactly the same. So this one, the only way that we know that it bleached previously is its tag. So we can actually go back to these corals now and know that this is the more resilient one. So that's the living library that we have to be able to show us which ones are more resistant to bleaching or more resilient. We can also do stress testing. So this is looking at respiration after a stress test. And that's this usually acute temperature stress that looks at the difference between different genotypes. So if you just go to a place and take some frags of corals, you can stress test them and see which genotypes are stronger, because we know now that the resilience is based on genotype. You will see from the same colony, they will act the same in these stress tests, and some genotypes are stronger than others. This is Eva here, and she didn't talk about it today, but she's also working on building some genetic markers where you could just take this tool to be able to go anywhere in the world and be able to test a piece of coral and say whether that was more resistant to bleaching or not. And then also we've been working on this collaboration with Greg Asner, who is now at Arizona State University, but they have a lot of collaboration. He's also on the Big Island. And you might have heard that there was just an article with him peeling away the seawater. I don't know if you saw that, but it's pretty cool. And they're now interested in a lot of Hawaii work as well. So you'll probably hear his name soon enough if you haven't already. But what we did is we mapped all of Kaneohe Bay using hyperspectral imagery. And we're not sure what the signal is yet, but we can tell, I think last I checked, it was like 75% accuracy at least, which ones bleached previously and which ones did not. So we're not sure why yet. It's not just the chlorophyll, it's probably a coupling of multiple different factors. But we can use all of these methods to tell us right now with what exists in the bay or wherever we're working, what exists, and tell us which ones are more resilient from those. So I'm going to focus mainly on stress testing. This is the one that we have the most well established. And as you can see, as you stress them, you can see that they fall out based on their bleaching. But again, we're not trying to kill them. We want them to be able to recover. So we're looking at the different genotypes here. And we can also, we just built a new system where we could actually mimic these bleaching events that Eva showed you this graph before. We can mimic those bleaching events and see if the corals behave similarly to that. So we have control and do the regular diurnal cycles. So we're not just instantly stressing them. We still follow the natural flow that we would just increase the temperature gradually. So with this, our new project that we just actually signed the agreement for a couple of weeks ago is to selectively propagate for restoration purposes using resilient corals. So if you know that you're selecting corals that are more resilient and using those for restoration efforts that you're already mandated to do, for example, for these ship groundings or boat groundings, then you're going to be increasing your return on investment and also enhancing future success, because the next time a warming event comes in, which might happen this summer, those corals would be more prepared to withstand that warming event and not get stressed, not bleach. So you have a better chance of all of your restoration efforts that you spent all this time and effort on and all this money, that they don't just get wiped out by a bleaching event. So this is just an example in Kaneohe Bay. This boat grounded right in front of a bunch of coral biologists. What are the chances? But unfortunately, it happens a lot. And you can see the destruction This is that reef. This is the same exact reef right here that we had a picture of before. And this is right after it happened. And it was only there for maybe an hour, just rocking back and forth. And I mean, these corals are thousands of years old. So this is really important for us as an island state, because one of the main reasons that we need to protect coral reefs is because they protect us. They break up the wave energy that comes in, you know, with sea level rise, things like that are occurring. We need to make sure that we have our natural barrier, which is our reefs, to be able to protect us. This is a really cool report that just came out. But these reefs are our reefs for us. They can dissipate up to 97% of wave energy. And just in Hawaii alone, it's protecting 836 million in infrastructure. And as you can see, all of us, I mean, especially here, I didn't really think about this until our funders mentioned it, but we only have one way in and out for emergencies. I grew up here. It didn't cross my mind, but we're kind of doomed when natural disasters happen. So our plan is we're partnering up with the state and also NOAA, who are already mandated to do restoration for things like those ship groundings, and also a nonprofit that I'll talk about in a little bit. And we are going to be working at 3 different sites: Kaneohe Bay, because we already have so much knowledge of the corals there; South Shore Airport, because NOAA is already working there, and then that helps us protect the airport, which is one of our main important areas. And Honolulu Bay, because the community has really been calling for it there. So we do want to be able to respond when the community does ask us for help. And then we can also incorporate the community and do that as a test run to see if we can scale up using community efforts. And then NOAA also just recently asked us to expand into the Pacific, so the USAPIs. And we're going to try to do some parallel work with them, similar to how we did in Australia. So let's see. I am going to skip through. This is just kind of, I'll just show you some pictures because I want to be able to have you guys ask some questions. But this is that community effort. We've already been doing a lot of that. As I said, we invite people in to help with spawning. But that's really the only way we're ever going to be able to scale up restoration. We're a small lab. DAR doesn't have many employees. No, like we need the community to get involved. And our main question as scientists for this restoration project is if resilient corals actually enhance restoration efforts, because we talked about those trade-offs. So we don't even know. We think it's going to work and that it's going to be beneficial. We don't know yet, so we're going to find that out. And this is just some pictures to show you. We're going to do some nurseries in the water. This one, that top one exists already out by the airport. And then this is our table that exists in Kaneohe Bay. We do fragmenting and microfragmenting. And then I just wanted to show this is coral growth within 6 months' time, which is actually a lot of growth for Hawaii corals. We're usually known for having really slow growing corals. And this is at the state. They're doing a lot of microfragging right now. And these are 3 locations that we've talked about. And we'll be out planning in plots. And we will look at random versus those that are resilient. We'll do regular monitoring, just like best practices have shown us. And the main thing is that we really are seeking the community's help. And it'll be in our 3rd year when we're moving over to Honolulu Bay and really trying out for best practices to see if we can have communities, especially in outer islands, be able to do this type of work. So our question, to bleach or not to bleach, it's up to all of us to determine that. So thank you very much for your time. I do want to give a just recognition to Ruth Gates, who had all of these great ideas and started this lab, and we're trying to continue on everything great that she's built. And now we'll take questions.
Any questions? Yes.
You mentioned several types of microalgae, different species, and are species associated with coral species, or is that the wrong way of asking?
No, that's great. So we call them clades and also types. So when I had that like C, D, is there? I'm not sure if I can see. When I went through C, D, those different ones, those are the clades, and then they each have a different type. So, oh, it's weird, it's not showing up. But this study right here, that paper showed they use this big incubator, and that one shows all the different clades and types and their thermal tolerance. So they tested them when they were free living to see how they responded to thermal stress, and then now it's trying to inoculate them into coral larvae and see, because I mean they might be thermally tolerant, but it might not actually benefit the coral. But yeah, so that's like D1, like 1 is the type, G3 is three is the type for that clade of G.
I guess part of my question is with clay on those species coral.
Yeah, so it's the coral that's really selective. So some of them you can put it in front of a coral and it won't take it up. They're very selective about what they take up. And then it's really interesting that in order to bring the algae into their cells, they actually they bring it through their stomach. So they pretty much eat it, but then they know that it's algae and they want to save it and they bring it into their cells instead of actually ingesting it. So yeah, corals are just so amazing. It's like every time you think you've understood something, there's another layer you have to investigate.
First of all, great information and great work, you guys. It's pretty amazing. Yeah. A couple of questions, and I might have missed this, but aside from preconditioning coral, is there such? And you spoke about pro-death inhibitors. Is there such or pro-life inhibitors? Is there such a thing as pro-death gene and box inhibitors? And could that be great?
Yeah, so this is more complicated to get because, as I was telling you, there is this apoptosis, or these pathways are also present in human bodies, and they are related. Like usually, these genes are somehow related in cancer. So people are really into developing everything that can stop cancer. So it's like stopping cancer means making cancer cells die. So all the effort of people in pharmacy and people who are developing drugs and inhibitors is to make cancer cells die. But I want to prevent coral cells from dying. So I go the opposite way. So there are almost no inhibitors on the market because there's no one who would make them. So that's more tricky question. That's why I can't answer this question if we can somehow mimic the preconditioning and prevent bleaching using drugs or whatever. I can't answer this question right now because I'm still working on methods how to go the other way that all the human cancer biologists and people go. Yeah.
And the second question is there a time frame associated with acclimatization or preconditioning and coral's ability to resist bleaching? Like X amount of time will last X amount of time of resilience.
Mm-hmm. That's what we believe, but we don't have exact answers to this yet.
It's exactly what we're answering this summer, actually. Yeah. So we just started preparing for that experiment, and we actually had to build a whole new system of temperature control tanks in order to do it at the scale that we wanted. So we're testing, I believe it's 5 different profiles to see the way that you acclimatize them. So, kind of it's acute or chronic conditioning where like if you just do it really rapidly versus if you do it over a long period of time of say 3 months, does that make a difference? And then how long does that memory last? Yeah, it's exactly what we're.
Yeah, I can only say that in the case of Poecilopora acuta, it lasts less than 1 year because the temperatures I'm using to acclimatize them, to precondition them, are usually temperatures that you can have in Kaneohe Bay in September-ish, like around this month. So, and I know that they can naturally acclimatize in the field because that's what I tested last summer. I was interested in this when I saw that the temperatures in the bay naturally go very close to what I use in our artificial tank system. I was curious to know if these corals are preconditioned. So I sampled them from the field, and yes, they were preconditioned. So it's something that happens annually. So I think that this preconditioning lasts less than, let's say, 9-8 months, but I can't give you an exact answer.
Okay,
now.
Thank you.
How did you manage your biosphere? How did you maintain a living marine environment?
That's a great question. So it has existed in the past, and it was kind of forgotten about for a while, or there wasn't a lot of work to be done there. So all of a sudden the corals that were there died off and all this algae came in. So we have a postdoc with our lab that's actually spending a lot of time out there right now, and he's working on shifting that tank back from an algae dominated system to where we can even put corals back in. So all these ideas that we have for the biosphere too, we're really excited to start, but we have to do the basics first of getting it. And but he's also he's doing research on microbes to look at what happens when you do shift the system from algal domination to corals. So yeah, it is a bit complex, and it's really, yeah, it's we're all going to go out there and get to scrub some algae at some point or another. But yeah, it's a little bit of a process, and he's a postdoc, so he's ready to get rolling with the big stuff, but he has to take care of this first.
Anyone else?
One more general question, I guess. Can you give one answer or range to the question? A healthy coral gains how much of its, what percentage of its nutrition from algae, what percentage from catching other critters with chemicals?
Good question.
Yeah, it's a very good question.
I can answer that. So we know that in some species, of course, it depends on species. Some species are very ultimately symbiotic, meaning that they can do as low as 10% by self-feeding, and they depend by 90% on the symbionts. But then we know species that can do much better, and we even know species, it's basically soft corals, not hard corals, but they can live without symbionts completely. And also we know some anemones that can do without symbionts. There is also like one anemone that can do both. So if there are symbionts that in the environment, they will catch the symbionts and they will live happily with the symbionts. But if you make them bleach, they will survive years without symbionts because they can make their own food for them.
And actually, one of our recent graduates, he just finished his PhD, Chris Wall, he looked at it in a different way. He looked at stable isotopes to be able to tell whether corals were heterotrophically feeding during a stressful event. And he surprisingly found that it didn't seem like they were actually choosing to feed when they were in that stressful situation, which is opposite of what we always thought. So it's really interesting, and more investigation needs to be done. And it might have just been that one species, or it's always. But it was really interesting to see that he didn't pick up a big signature for that.